Hand editing of Ccrs out of CD4 cells of persons infected with HIV. Definition. CD4 is the mejor coverage for human immunization virus HIV with investing with the modification of the gene. In this case, the infusion of the CD4 the cell in which they 5 gangwags render permanent diffusion by anti-French nucleic.
So that they can start to use that to develop their own and design their own experiments with the gene that they're interested in being changed in whatever way they want. So what they're trying to do is they're trying to see if they can change blood cells. So they can take blood cells from a patient, they can change the gene in there, and they can put it back into a patient. And one of the genes that they're trying to change is called Ccr5, and what they're trying to do is trying to change that gene so that HIV can no longer infect these cells, because Ccr5 is what actually helps the HIV virus enter into the cell. Take the cells from the blood, change the gene, put it back in the blood, and they can put it back in the patient. They would then be resistant to HIV, which would be phenomenal.
Macduff. We are love part in and by life more life. Un goal is to effort English dove modifying out of CD4 themselves days. Passing up challenge average achieve infection will they rising in his activate that part.
Drug treatment a few hours after birth. It is the second case of its kind.
Meanwhile, a new study focuses on a promising therapy called gene editing. Researchers are using it to create immunity to the virus that causes AIDS. Dr. John Lapuka is with us this morning. Good morning.
Hey, Charlie.
Explain this, how it works and how significant it is.
This is a technological tour de force out of the University of Pennsylvania. And it's based on the fact that an HIV virus enters a white cell and infects it, a T cell, by a protein that's on the surface of that cell, okay? Without that protein, it cannot get into the cell. It's locked out. And what these researchers did was they took the white cells from somebody with HIV, they brought them outside the body, and then imagine this. Out of 20,000 genes that are in a cell, they found the one that controls for that protein on the cell surface. Yeah. They disabled it, they put them back into the person, and now the person had the HIV virus circulating, they couldn't get into the cells to infect it.
I think this is fascinating, right? Because this is the future of medicine, is gene therapy and genetics. And in this specific case with HIV, it was this mutation called CCR5, that if you inherited that from both parents, it's very rare, you wouldn't get HIV. So scientists found that and said, let's make this happen. How successful is it? Does it cure you of HIV?
Well, they're not using the word cure, but this is a small safety study, okay? And there was 12 patients, 6 of them they could really look at because they could stop the medication and see what happened. In 4 of those, the HIV levels went down. 1 of those 4, it actually was undetectable.
All right, thank you.
This is really, I agree with her, extraordinary stuff. And in fact, there was only one gene, which makes it much easier.
That's definitely.
Thank you, John.
Next, GP120 undergoes a conformational change, which allows it to bind to a second receptor, or co-receptor, on the host cell surface. Binding to the co-receptor is critical to the virus, as without this event, HIV cannot enter the cell. Finally, co-receptor binding induces structural changes in another viral glycoprotein, known as GP41, which result in fusion of HIV with the host cell and allows entry of the virus.
results. Unsure adverse event was associated with infused antigens, modifying adult CD4 cells and was identified for transfusion reaction. They median CD4 they cell account was very good Miller 1 and siblings in gram. For they presume a cook of per cubic millimeter days median concert fund modified CD4 they cell and was cell per millage the constitute and circle per wall monitor self.
Successful replication and is therefore a potential target of antiretroviral drugs. Step 1 is the infection of a suitable host cell, such as a CD4 positive T lymphocyte. Entry of HIV into the cell requires the presence of certain receptors on the cell surface. CD4 receptors and co-receptors, such as CCR5 or CXCR4. These receptors interact with protein complexes, which are embedded in the viral envelope. These complexes are composed of two glycoproteins, an extracellular GP120 and a transmembrane GP41. When HIV approaches a target cell, GP120 binds to the CD4 receptors. This process is termed attachment. It promotes further binding to a co-receptor. Co-receptor binding results in a conformational change in GP120. This allows GP41 to unfold and insert its hydrophobic terminus into the cell membrane. GP41 then folds back on itself. This draws the virus towards the cell and facilitates the fusion of their membranes.
Unsure adverse and was association with infused modified adult CD4 for this cell and wax to transfusion that is CD4 T cell and for previous Miller 1 a significant and from the presupport per cubic meter day median concentration modify CD4 cell and 1.
The co-receptor for HIV. Why is that a good target? It's a cellular gene. Don't we need it? If you knock it out, won't we lose important function? Well, it happens that there is a natural population of people who carry 2 mutant copies of the gene for CCR5 and can't make any CCR5. So their cells have no CCR5 on their surface. Those people are actually resistant to HIV infection, because CCR5 is an absolute requirement for HIV infection. But amazingly, they have virtually no other immune defects. They may have defects in relation to one or another specific organism, but to a first approximation, people live a normal life without this gene. So if we can knock down this gene in the cells of an HIV-infected person, those cells will now be able to grow up and replace helper T cell function. Now, some people carry 1 mutant gene and 1 normal gene. Those people make somewhat less than 50% of the normal amount of CCR5, and they develop AIDS much more slowly than normal people do. And so we know that even if we could just knock it down 50 or 60%, we could actually affect the course of the disease. If we can knock it down 95%, we can probably have a major effect. So what we're trying to do is to bring into helper T cells an interfering RNA. It's called an RNAi or an siRNA or an shRNA variously, which is able to block the translation of the messenger RNA for CCR5 or cause degradation of the messenger RNA for CCR5. And thus, it'll act like that mutation and protect those cells and those people.